Okay, Vivek, right? Okay.
Yes, yes, sir.
So, Vivek, just introduce yourself in brief.
Yes, sir. Sir, my name is Vivek Kumar. I did my graduation in 2014 in biotechnology. Following that, I completed my master's in again in biotechnology. Following that, I got a, job placement from campus only. So I taught for 3.5 years. That is 2016 to 2022. Sorry, 2020. So during corona, I lost my job, but during that time, a vacancy was there for joining technical assistant in lab at Bureau of Indian Standard. So I joined Bureau of Indian Standard on the post of technical assistant lab in the year 2021, and till now I am working there.
Okay, so currently you are working in Indian Standard Laboratory as a technical assistant.
Yes, sir.
Okay, well done, very good. So, Vivek, what is your area of interest?
So my area of interest is biochemistry.
Biochemistry. Okay, very good, well done. So, may I ask some questions to you regarding
Yes,
biochemistry?
yes, sir, you may ask, sir.
Okay, so my first question is that why you like more biochemistry as compared to any other subject?
So, biochemistry, if you look at the other subject, and if you go in depth in other subject, we come across with the biochemistry pathway. So either it is immunology, molecular biology. If you go at depth, everywhere is some biochemical pathway is going on. So that's attracted me a bit that biochemistry, we should know biochemistry. That's why it becomes my favorite subject.
Okay, means basically you are interested in chemical kind of reactions. Okay,
Yes, sir, yes, sir, yes, sir.
okay. As you earlier said that you have also experience of teaching some 2 or 3 years.
Yes, sir, yes, sir,
Suppose
yes, sir.
suppose I am your student and you are a teacher of biochemistry and I have no any interest in biochemistry. So being a teacher, what will you do to develop interest in me regarding biochemistry? What will be your steps? How you can develop interest in your subject toward in the students' minds?
So before starting biochemistry and before making student liking this subject very much, uh, I will start. I will teach him from basics of chemistry, which is required, which is required to understand the biochemistry. So if I make the student feel better, more confident in basics of chemistry, then that student will automatically start liking biochemistry. In that way, I also like to guide the student starting from basics of chemistry. Then they will able to pick up the biochemistry and start liking that.
Okay, okay, okay. So to be a good in biochemistry, there is a need of a concept of a biochemistry itself, right?
The basics of chemistry should be there, sir.
Okay, so Vivek, in life science, especially in biochemistry, there are biological interactions, right? There are
Yes,
certain
sir.
kind of a chemical bonds occur in the living systems. Can you
Yes, sir.
suggest some chemical bond which is very very important in biological system?
Like covalent bond, that is a peptide bond occurring in the entire protein biochemistry. Without peptide bond, amino acid cannot link and protein cannot be generated. Apart from that, we will looking towards the glycoside bond, which are forming during the cyclization of glucose molecule, fructose molecules. In also in nucleotides, there is some glycosidic bond that is very beneficial for that molecules. Apart from that, um, there are some non-covalent interactions are also there, like hydrogen bond, van der Waals interactions, hydrophobic interaction. These are very essential for folding of protein from primary to going towards quaternary structure. These kind of interactions are very important.
Okay, hydrogen bond. How you
Yes,
look
sir.
about the hydrogen bond in biochemistry? What are the basic role of these hydrogen bond in biochemistry?
So, if you look at the protein chemistry, uh, to fold up to get functionality in protein, primary structure of protein must be folded into secondary structure, then tertiary, then quaternary structure. So in order to fold the primary structure into secondary structure, hydrogen bond is very important. So the hydrogen bond link one amino acid to the other amino acid to get in a helical manner to generate the secondary structure like alpha helix, beta plate structure of protein. So there the hydrogen bond plays a very important role. Apart from that, water molecules are there in the living system. So lot of biochemical reactions are occurring there. There also hydrogen bond is very important to link water molecules with the other functional groups. So hydrogen bond has some importance there in the biochemistry.
Okay, hydrogen bond. Is it a real bond or something different?
So it is a real bond. So hydrogen bond forms between two electronegative atoms. If you take example of water, in water hydrogen bond will form between oxygen of one water molecules and oxygen of other water molecules. So it is a real bond that will attract the two water molecules to come together.
Okay, Vivek, how we can decide and how we can define what is the real bond? What is the criteria to define real bond?
Sorry, sir, I haven't read about that how to define.
Okay. Suppose you have a chemical bond. Chemical bond means
Mhm.
that is there is a involvement of electron.
Yes, sir.
So on the basis of that, how you look about the hydrogen bond? Hydrogen bond is a real bond, or just like it's a interactions like a hydrophobic, hydrophilic, etc.
So we can say that it is a kind of attraction between two electronegativity atom. There is no such transfer of electrons from one molecule to another. It is just attraction.
Then how
So
you can say like that is the hydrogen bond is a real bond?
yeah, yes, in that manner we cannot say it is a real one. Yes, sir.
Okay. Does each elements can form hydrogen bond, or there are some criteria to form hydrogen bonds?
So each element cannot form. So there should be some electronegativity. Means there should be some electronegative atom is there, which had some differences in electronegativity, which may attract hydrogen. Like oxygen is there, nitrogen is there, fluorine is there. We cannot form hydrogen bond between two carbon atoms because it does not have that kind of electronegativity uh, capacity which can attract hydrogen to
Okay,
form the
Vivek,
bond.
except all these three, that is that is oxygen, nitrogen, and fluorine. Does any other elements can form hydrogen bond in the all the periodic table?
So, sorry, sir, I cannot recall that if any atom because I have read about these three atoms most frequently in chemistry and also in biochemistry. That's why
Okay,
I remember.
water has a very significant role in biochemistry, right?
Yes,
So,
sir.
can you elaborate some properties of the water molecule on the basis of a chemistry?
So it has good.
What is the geometry of water molecule?
So it's a vent structure, V-shaped structure, and and the bond angle is 104.5 degrees. 5 degrees is the bond angle. V-shaped structure is there, and there are two lone pair of electrons present in oxygen atom. So that's the structure of water molecules.
In terms of Vesper theory, that is either it is trigonal, tetrahedral, hexagonal, square. What is the structure of a water molecule on the basis of Vesper theory?
So on the basis of Vesper theory, so I cannot remember, sir,
Okay,
because
what is the hybridization of oxygen in water molecule?
sp3 hybridization, sir.
sp3.
sp3.
So
Yes,
if
sir.
any molecule have sp3 hybridization, then what will be the structure of that molecule?
Yes, sir. In that case, it is a tetrahedral structure is there, and shape maybe like pyramidal shape because two lone pair of electrons are there. That will make the pyramidal shape, and the structure is tetrahedral.
So does water molecule have tetrahedral structure?
Yes, according to hybridization, it is tetrahedral structure, sir.
Have you heard about the distorted geometry?
Yes, sir, I heard about distorted
So what
geometry.
is the distorted tetrahedral?
So distorted, it means, sir, water molecules should be there because there are two lone pair of electrons. It will repel each other. So that may be regarded as distorted tetrahedral structure.
Okay, Vivek, tell me one thing. In pH scale, there are values from 0 to 1, 0 to 14,
Yes,
right?
sir. Yes, sir.
So, is there any other possibility that that scale will move onward, that is 15, 16, 17, etc.? Why it is just only 14, not 13, 12, or 15, 16?
So it has been standardized between 0 to 14 only.
Why? Why the
So because,
pH value have a scale, pH scale have value that is 0, 0 to 14, not other than 20, 15, 16, 17, etc.?
sir, most sir, in biological system, most pH value of blood is 1.5, and in other also, sir, pH ranges
pH scale
from.
is not limited to the biological system. pH scales can
Yes,
be
sir.
used in everywhere, for the soil, for water,
Yes,
for
sir.
any other
Yes, sir.
fruit juice, etc.
Yes, sir.
So pH scale is not the concept of the biological system. It's a basically concept of chemistry. And in
Yes, sir.
chemistry,
Yes,
there
sir.
are so many chemicals, and there are so variations of the pH value. So why we have
Yes, sir.
only 0 to 14 range, value of range in the pH meter, pH scale?
Sorry, sir, I haven't read that why it has been fixed between 0 to
Okay, so
14.
being a biochemistry student, you must thought about that.
Okay, sir.
Okay, definitely you will think and you will find some good answers. And at the end of interview, I will explain each and every questions.
Okay, sir.
Okay,
Okay, sir.
so moving forward, what is isoelectric point?
Sir, it is that pH at which molecule has a net zero charge. Means it is electrically neutral.
Okay, generally we state that water is very necessary for life.
Yes, sir.
Life cannot be possible without water. Can you justify the significance of water in biological system? How water is so significant for the survival of the any living system?
So, I can say that, like, sir, during when woman gets pregnant, so if you look at the baby, it's still growing inside the water bag that is full of water and other nutrients in there. So that signifies that water is very essential for the life. Your life begins from water only. So that I can say that. Apart from that, our body contains 70 to 80% of water, and in water, a lot of reactions are going on. So that also signifies that water has an important role.
Okay, Vivek, egg, that is white egg, has a calcium carbonate cover,
Yes,
and
sir.
inside the calcium carbonate cover there is a yellowish liquid. When the egg will boil, then all the liquids will disappear and the material will solidify.
Yes, sir.
As you know, calcium carbonate is just like a stone, and there is a no porosity, there is no chances to evaporate the water molecule from inside to outside. So what happened with the water molecules, where it will go after boiling? The egg become solidifying.
Yes, sir.
So where the water molecules evaporated? What is the chemistry behind it?
So, egg,
Are you
egg
getting
white.
my question?
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
ओके, मेरा सवाल ये है कि अंडा जो कि आपका लिक्विड है, आपने उसको
Yes, sir.
बॉयल किया, सो डेफिनेटली वो क्या हो गया, पूरा सॉलिडिफाई हो गया। कैल्शियम
Yes,
कार्बोनेट
sir.
की जो लेयर है वो आपका, उसमें कोई पोरोसिटी नहीं है। सो हाउ इट इज़ पॉसिबल कि वाटर मॉलिक्यूल गया कहाँ, कैसे सॉलिडिफाई हो गया?
सर, वहाँ सर प्रोटीन का डिनेचुरेशन हुआ होगा सर, एंड इन दैट डिनेचुरेशन वाटर में बी लॉस्ट समवेयर देयर ओनली। सर, मैंने पढ़ा था इसके बारे में कहीं बहुत पहले सर, आई कैन नॉट रिकॉल। वहाँ एक केमिस्ट्री होती है कि कुछ प्रोटीन का डिनेचुरेशन हो जाता है, और पानी वहाँ कहीं पर एब्जॉर्ब हो जाता होगा। but I
Okay.
cannot
In metabolism or bioenergetics, there are a concept of the thermodynamics also. So, what is the mean of enthalpy?
so enthalpy is the total heat content of the given reactions, which is indicated by del H.
So, on the basis of enthalpy, biochemical reactions can be categorized into two classes. What are these?
Endothermic and exothermic reactions.
Exothermic. And what is exergonic and endergonic?
So I think so both are synonymous.
Both are same. Then why if we use these two different terms, that is exergonic, endothermic, endothermic, endergonic?
So I can recall the interzonic and exozonic. It may be on the basis of favorable reactions or disfavorable reaction, and endothermic and exothermic is based on the, in case of endothermic reactions, heat are being absorbed, and in case of exothermic reaction, heat are being released. So on the basis of that, it has been categorized as endothermic and exothermic. But in case of interzonic and exerzonic, I think, sir, this is all about favorable reactions and non-favorable reactions. Like del G is negative. Sorry, sir, I cannot recall all
Okay,
the word.
suppose you are making a sugar and water solution. You are dissolving sugar crystals in water molecule. Which kind of reactions are there? Exothermic, endothermic, exergonic, endergonic. What kind of reactions are there?
So it's an endothermic reaction, sir, because after dissolving glucose inside the water, sorry, sir,
Endothermic, that means the heat will required.
no, sir, heat has been released. Sorry, sir, I cannot recall what is happening.
Okay, what is entropy?
So entropy, so it is the disorderness of the system. Like some reaction is going on, so what is the total disorderness of the reaction? Right? So it can be said in terms of that if more molecules are forming, it means it is more disorder. If less molecules form, it is less disorder. In that manner, it is defined as what is entropy.
Suppose you have a four different level of a protein structures like the primary, secondary, tertiary, and quaternary. So, in different level of a protein structures, which level of structures has highest entropy and why?
Sorry, sir, I haven't read about the entropy which is going from primary to quaternary level.
Okay, okay. Suppose you are dealing with a protein and that protein has a pi value that is 6.5. Okay, and if the pH of that media is 6.8, so what will be the overall charge on that protein? The pi value is 6.7 and the pH value is 6.8. What will be the overall charge on that protein?
So since pH value is 6.8, pI is 6.7, means environment is getting more alkaline. So in that case, uh,
Suppose it can be arranged like as a, suppose pH value has a 6 pH and the solutions of that protein is 8 pH. So what will be the overall charge on that protein?
so it will becomes a negative charge because OH will get deprotonated and ammonia group is already protonated. So after getting deprotonated, it will become neutral, but there is charge on the charge on the oxygen group. So it will become overall neutral, overall negative charge.
Overall negative charge.
Okay.
What is deuterion?
Such deuterions, sir, both positive and negative charge is there on the deuterions. Like NH3 is also protonated, and the OH is deprotonated, their negative charge. So overall charge is neutral, but there is positive and negative charge in equal amount.
So is there any differences in deuterion and isoelectric point?
Uh, no, sir, there is no difference because deuterions itself occurs at pi, means at that pH, at which their pI becomes equal to that pH. So deuterium is same as the pI, sir.
Okay, Vivek, have you heard about the 2D electrophoresis?
Yes, I heard about
Okay,
2D
what is this?
electrophoresis. So in 2D electrophoresis, proteins are separated on the basis of on the basis of their respective pI value.
Then there is also a separate technique that is isoelectric foxing, IEF. On the
Yes, sir.
for the
Yes,
separation
sir.
of a
Yes, sir.
protein on the basis of isoelectric point, there is a technique
Yes,
that
sir.
is known as the isoelectric foxing. Then why we use
Yes, sir.
2D electrophoresis?
Oh, sorry, I remember, sir. In 2D electrophoresis, there are two parameters on which proteins are separated. One parameter is on the basis of pI value, and second parameter is on the basis of their relative molecular weight.
Why it is known as 2D? Is there any concept of 1D?
So because there is proteins are being separated in two dimensions. One is the pI value, other is the relative molecular weight value. That is why it is two dimensional.
Well,
That is why it is.
very good. What is a spectroscopy?
Sir, a spectroscopy is it is the interaction of molecules with the given wavelength of light.
Different wavelength of light.
Yes, sir.
That means electromagnetic radiations.
So it should be monochromatic light.
Only monochromatic light.
Yes, sir.
Then
Only
how we
some
can use UV visible spectroscopy, because there are the range of the certain range of the wavelength, and monochromatic light means that is a light having single wavelength.
yes, sir. Yes, sir.
Then how we can
No,
use UV visible spectroscopy?
sorry, sir. Sir. In a spectroscope, there is a range of wavelength, and we have to select at a
So how you
particular
define
wavelength.
spectroscopy?
So you can say that it is the interaction of given molecules with the electromagnetic radiations,
Good.
which is
Then what is the electromagnetic radiations?
so electromagnetic radiation. It is a spectrum of light where various various light of different wavelengths are there, like X-rays, gamma rays, UV rays, and visual spectrum there. So all
Then
falls
what
in
is the
electromagnetic.
spectrum? If these things are the electromagnetic radiation, then what is the spectrum?
So a spectrum, so it is a series of light, which is the series of light at various wavelength. So that generates a spectrum.
Or what is electromagnetic radiations?
So electromagnetic radiation, so there is certain characteristics like the light interacts both with the electric field and the magnetic field. So that's why it has given the name electromagnetic radiation.
Does it require any material medium for their traveling?
No, so it do not require any material medium.
How you can prove, Vivek, that electromagnetic radiations do not require any material medium for their traveling? How you can prove it?
So, like mm, during sunlight, light energy comes from suns towards Earth. It may be traveling through a space. In a space, there is no medium. So, so it may prove that light do not require any medium to travel.
Okay, Vivek, suppose you are traveling in a that is aeroplane, and the height of that aeroplane is above to the our atmospheric region, and you just looked that is the sky. What will be the color of the sky?
So orange color.
Orange color. So why?
So it may be according to the wavelength of the light which is coming from the sun.
Why sky appears blue?
So, sorry, due to a scattering of blue light.
So for the scattering of light, what is the conditions? How? Why? In which conditions light will be scattered?
Sorry, sir, I cannot remember the concept of scattering. I just have read that.
Okay. Have you heard about the absorption spectroscopy?
Yes, I heard about absorption
What
spectroscopy.
is absorption spectroscopy?
So in absorption spectroscopy, light of certain wavelength, when light interacts with given molecules, some lights are absorbed and some lights are transmitted to the system. So the light which has been absorbed by the molecules, that will be detected in the form of transmitted light, which moves to the uh, uh, to the machine. That machine will detect the transmitted light. So that's the function of absorption spectroscopy.
Have you heard about the NMR, ESR spectroscopy, Raman spectroscopy?
Yes, a little bit I heard about that.
Okay, so can you say something about the NMR spectroscopy? Why we can use and in which conditions we can use NMR spectroscopy?
So NMR in biological system, NMR spectroscopy is used to analyze the analyze the protein molecules. Mainly, sir, their three-dimensional structure. Also, in chemistry, we use NMR structure to identify the functional group of a given compound. So that is the function of NMR.
Okay, side exclusion chromatography is one of the most important technique in the biochemistry, which is used for the separation of protein molecules.
Yes, sir.
Can you explain the mechanism or the working principle of a size exclusion chromatography? How
So
the size
size
exclusion chromatography separate the different shape of the protein molecules?
size exclusion chromatography works on the principle of separation of protein mixture of protein molecules on the basis of their relative molecular weight. So
Relative
during
molecular
preparation,
weight, not on the size. Then why it is known as a size exclusion?
so we say relative molecular weight or size. So I think both are
If the working principle of a size exclusion chromatography is based on the molecular weight, then why it is known as a size exclusion chromatography?
so weight may be considered as a size. So if
There are
some molecular,
there are so many examples that the size is lesser, but the molecular weight is heavier. Suppose you
yes,
have a cotton and you have a mercury,
yes,
the same shape will be more higher.
yes.
So
So
yeah, go ahead.
sorry, sir, I used the wrong term. So it works on the basis of size. During preparation of proteins, first of all, we have to neutralize the charges given on the mixture of proteins. After neutralizing the charges, the protein which are having very small size, it will come at later stage because it has to travel along all the pore size which is uh, which is in the bid. So it has to travel long distance to come out of the molecules. It will come at last. The protein which are of higher size, it will comes out of the bid, and it will appears in the solution at first. So higher size of protein comes first, and the lowest protein size comes later, and in between all the protein falls according to the size.
Why this happens? Why smaller size molecules will elevate later and larger molecules will elute first? Why this happen?
So because the size of the smallest protein is smaller than the bit size. So bid size is larger, so it will get into the bid. There are thousands of bids are there in the stationary phase of chromatography. So it has to travel all along the bid, then it will come outside. And the larger size of protein, its size is larger than the bid size. That's why it do not get penetrated inside the bid. So it will come first. It will come outside of the chromatography at first, taking very short distance.
Okay. So what is affinity chromatography?
So affinity chromatography, it works on the basis of uh, affinity of the reagent which is attached on the bid with the affinity molecules yeah, with the affinity molecules present on the given biological molecule. So there is some affinity should be there. Like I can say that if you want to elute the eukaryotic mRNA, which is so which character is that it is poly A adenylated. So in order to elute the mRNA molecule, you have to tag poly T on the bit side. So that poly T will interact with the poly A of the mRNA, and in doing that, its affinity is there. It will take out, it will bind the mRNA molecules. So after that, if we increase the salt concentration, that affinity breaks down and mRNA comes out of the solutions, leaving behind other solutions which do not have any affinity. That way, the affinity chromatography works.
Okay, Vivek's, there are different kinds of a chromatography technique, like as a affinity chromatography, like as a size string chromatography, like as a ionization chromatography, differently, right? Suppose you want to uh, isolate or filtrate enzyme molecules from a sample solutions. So among these three chromatography technique, which technique you prefer to use for the enzyme separation, and why?
Sorry, sir, I cannot say that. I am not able to recall about the how will you separate. So I cannot say that. Sorry, sir.
Okay, suppose you have a solution and there are certain molecules of the protein bearing negative charge, and you want to just separate out that negatively charged protein molecules from the mixture of the solution, from the mixture of the protein, right? So which kind of a technique you will use for the separation of negatively charged?
We will use the ion chromatography, sir.
Which one? Iron has two
Sir,
types, that is cations and anions.
okay, yes, sir. Sir, I will use the anion exchanger, sir.
An ion exchanger. Okay, why?
So because we have the negative charge protein, so that will be exchanged with another negative charge molecules. So it will go and attach with the anion exchanger, and other anion molecule will come out and it will go and bind there.
So what is the charge of the bead used in the anion exchanger?
Suppose so positive charge is there on the anion exchanger. It
Then why
will
it is known as anion exchanger? Positively charged particles known as the cation. If
yes, sir.
if the anion exchanger has a positively charged bead, then why it is known as the anion exchanger?
So because it is exchanging the because it is exchanging the molecule which has negative charge. That's why it is said that it is anion exchanger, exchanging anions, negative charge anions molecule with other negative anions molecule. That's why it is said anion exchanger. Similarly, there is a cation exchanger also there. It is exchanging one cation molecule with the other cation molecules.
Okay. What is centrifugation?
So centrifugation. So so it's a centrifugation. Sorry, sir, I cannot able to recall the definitions of
Centrifugation is a process or any instrument.
centrifugation is a process. It is done by it is done by uh, centrifuge machine. So in centrifuge machine, using centrifugal force, different different mixture of different molecules are separated on the basis of their density. Higher denser molecule will go and form pellet at the bottom of the tube, and the lighter one will come at the at the on the at the surface of the supernatant. So in that way, sir, it works using the centrifugal force.
Okay, Vivek, have you heard about the SPLC?
Yes, I heard about
What
HPLC,
is the
sir.
working principle of SPLC and how it is
So,
different from the normal chromatography, liquid chromatography?
so in HPLC, there is no there is two mode of working. One is normal phase, other is reverse phase. So I cannot exactly remember, but I can say that in one of the phase, a stationary phase is polar, and and the liquid and the mobile phase is non-polar. and in another phase, a stationary phase is non-polar, mobile phase is polar. But I cannot exactly remember in which this happened, either in normal phase or reverse phase. So that is the two mode of working, sir.
Okay, thank you, Vivek. Thank you for your nice interactions and session. Now the session is
Thank
open
you,
for
sir.
you. If you have any doubt, any query, any questions or suggestions, etc., regarding your interview, so you can ask to me. And now you can feel comfortable and also talk in Hindi. ओके
Yes,
सो
sir.
उन सारे सवालों का जवाब मैं आपको दे दूंगा जिन जो सवाल मैंने आपसे पूछे हैं अगर आपके उनके
Mhm.
आपको उनका आंसर्स नहीं मिल पाता है तो और अगर आपका कोई सवाल है तो आप पहले पूछ लीजिए दिन इसके बाद हम उन सवालों पर डिस्कस करेंगे या बेटर होगा कि पहले मैं उन सवालों का आंसर्स आपको कर दूं जैसे मैंने आपसे पूछा था स्टार्टिंग में आप बता पाएंगे कि मैंने क्या पूछा था
सब दिमाग से भूल गया सर, याद नहीं आ
ओके
रहा।
पहला पहला मेरा सवाल था आपसे केमिकल बॉन्ड को लेकर के कि
हाँ सर, हाँ हाँ।
हाइड्रोजन बॉन्ड को आप क्या कोई एक रियल बॉन्ड बोलेंगे कि नहीं बोलेंगे तो
हम्म,
बेसिकली हाइड्रोजन बॉन्ड क्या है कि एक जैसे आपने एग्जांपल दिया था वाटर मॉलिक्यूल का राइट
हम्म,
वाटर मॉलिक्यूल में ऑक्सीजन से हाइड्रोजन का क्या है कोवलेंट बॉन्ड है एक मॉलिक्यूल के बीच में just
हम्म,
because of the electronegativity difference in the water I mean the oxygen and hydrogen there is a separation of electron so
yes,
electron
sir.
will move towards the oxygen and that's why there will be a partial negative charge
Yes,
while
sir.
proton that means hydrogen contain partial positive charge so another molecule of water oxygen bears partial negative charge and hydrogen bears partial positive charge so
Yes, sir.
hydrogen bond is the interactions between these two charges, that is partial positive charge of hydrogen and partial negative charge of the oxygen. So because
Yes,
there
sir.
is no involvement of any electrons, there is no formation of any chemical bond. That is why we can say that hydrogen bond is
Mhm.
just like an interaction. It is not a real chemical bond. And
Mhm. Thank you, sir.
there is a very significant of hydrogen bond in the biological system because in antigen antibody interactions, there is involvement of hydrogen bond. In hormones and receptors, there is involvement of hydrogen bond. In enzyme substrate reactions interactions, there is involvement of hydrogen bond. In protein structures, there is involvement of hydrogen bond. In plasma membrane, there is involvement of hydrogen bond. So there are so many cases where you can look that is hydrogen bonds are involved in the formation of such kind of interactions. So on the
Yes,
basis
sir.
of that, we can say that hydrogen bond is one of the non-covalent bond which play very significant role in biological system.
Yes, sir.
दूसरा सवाल मैंने आपसे पूछा था कि वाटर मॉलिक्यूल की ज्योमेट्री क्या होती है वाटर मॉलिक्यूल में हाइड्रेशन ऑफ इनका क्या होगा सो आपने आंसर्स उसका किया था कि वाटर मॉलिक्यूल में ऑक्सीजन की हाइब्रिडाइजेशन sp3 होगी और जैसा कि आपको मालूम है कि अगर sp3 हाइब्रिडाइजेशन होगा तो ज्योमेट्री टेट्राहेड्रल होना चाहिए लेकिन
Yes, sir.
चूंकि यहाँ पे ऑक्सीजन के पास दो लोन पेयर ऑफ इलेक्ट्रॉन है लोन पेयर में रिपल्सन्स बहुत ज़्यादा होता है जैसा कि वेस्पर थ्योरी आपको बताती है
Yes, sir.
लोन पेयर बॉन्ड पेयर बॉन्ड पेयर बॉन्ड पेयर तीन कैटेगरी के
Mhm.
इलेक्ट्रॉन्स हैं सबसे हाईएस्ट जो रिपल्सन होगा वो क्या करेगा लोन पेयर में होगा उसके बाद लोन पेयर बॉन्ड पेयर में उसके बाद बॉन्ड पेयर में वेस्पर थ्योरी
Yes,
हमें ये
sir.
बताती है कि किसी भी एलिमेंट के जो इलेक्ट्रॉन्स बॉन्ड में पार्टिसिपेट कर रहे हैं वो उस मैक्सिमम डिस्टेंस पर जाकर क्या करते हैं लाई करते हैं जहाँ से रिपल्सन क्या हो जाए जीरो हो जाए सो
Mhm.
ऑक्सीजन के पास चूंकि दो लोन पेयर ऑफ इलेक्ट्रॉन है दैट इज वाई यहाँ पे रिपल्सन होगी तो ज्योमेट्री आपकी नॉर्मल नहीं होती इट्स अ डिस्टॉर्टेड टेट्राहेड्रल दैट इज वाई
Yes,
देयर
sir.
इज अ स्लाइटली डिफरेंस इन द बॉन्ड एंगल
Yes, sir. Yes, sir.
नॉर्मल
Yes, sir.
टेट्राहेड्रल बॉन्ड एंगल दूसरा होगा वाटर मॉलिक्यूल केस का टेट्राहेड्रल का बॉन्ड एंगल क्या होगा दूसरा होगा
Mhm.
ओके देन मैंने आपसे पूछा था 2D इलेक्ट्रोफोरेसिस के बारे में तो आपने बताया उसका आंसर आपका सही था कि यहाँ पे दो पैरामीटर्स होंगे दो डायमेंशन होगा वन डायमेंशन में आप मॉलिकुलर बेसिस पे मॉलिकुलर वेट के बेसिस पे करेंगे और दूसरा क्या होगा आइसोलेटिक पॉइंट के बेसिस पे होगा दैट इज वाई सपोज अगर कोई दो प्रोटीन है जिनका मॉलिकुलर वेट अगर सेम होगा तो आइसोलेटिक पॉइंट के बेसिस सेपरेट होंगे और अगर उनका आइसोलेटिक पॉइंट भी सेम हुआ तो मॉलिकुलर बेस के बेसिस पे क्या होंगे सेपरेट होंगे ओके
Yes, sir. Yes, sir.
और क्या सवाल पूछा था मैंने याद है आपको कुछ
सर जैसे कि सर एक सवाल आपसे पूछा कि एंजाइम को सेपरेट करने के लिए कौन सा
वेरी गुड
टेक्निक।
आपसे मैंने पूछा था एफिनिटी क्रोमेटोग्राफी क्या होती है तो आपने बताया कि एफिनिटी क्रोमेटोग्राफी एक ऐसा टेक्निक है जहाँ पे एक क्या होता है लिगेंड मॉलिक्यूल होता है और इसका कॉम्प्लीमेंट्री मॉलिक्यूल क्या करता है उसमें जाकर के बाइंड कर देता है राइट सो जब हम
Yeah.
एफिनिटी क्रोमेटोग्राफी को पढ़ते हैं तो जो उसका कॉलम होता है उस कॉलम में क्या लगे होते हैं, बीट्स लगे हुए होते हैं जो कि लिगेंड के फॉर्म में होते हैं, और वो लिगेंड क्या करता है, उसके कॉम्प्लीमेंट्री स्ट्रक्चर का कोई स्ट्रक्चर अगर मिलेगा तो क्या करेगा, उसको बाइंड कर लेगा. So for the
Okay.
isolation of enzymes, for the isolation of antibody, for the isolation of hormones also we will use affinity chromatography, because all these molecules have a binding affinity with a specific shape of molecules.
Yes, yes, I got it, sir. I got it.
Okay.
हाँ
Next
सर,
what,
समझ गया
और क्या
सर.
था सवाल आपसे?
सर कुछ सर स्पेक्ट्रोस्कोपी से रिलेटेड आपने कुछ पूछा
Yeah,
था.
spectroscopy is basically interaction of electromagnetic radiation with the matter, simply, right?
यस सर.
And
यस सर.
electromagnetic radiations means that is the part of a radiation or the kind of radiations which do not require any material medium for their travel. ऐसा रेडिएशन जो कि अपने आप को ट्रेवल करने के लिए किसी मटेरियल मीडियम की रिक्वायरमेंट ना हो, उसे आप क्या बोलेंगे, ई इलेक्ट्रोमैग्नेटिक रेडिएशंस, ईएमआर.
यस सर.
Electromagnetic radiations contain different kinds of wavelength of light, and the graphical representation of that
यस
different
सर.
wavelength light in order of increasing order of wavelength or decreasing order of frequency is known as the spectrum.
ओके सर. ओके सर.
इलेक्ट्रोमैग्नेटिक रेडिएशंस में जितने भी डिफरेंट टाइप्स के रेडिएशन वेवलेंथ हैं, उनको वेवलेंथ के इंक्रीजिंग ऑर्डर में अरेंज किया जाए या फ्रीक्वेंसी के डिक्रीजिंग ऑर्डर में अरेंज किया जाए, तो उसको आप क्या बोलते हैं, स्पेक्ट्रम बोलते हैं.
यस सर.
और कुछ?
यस सर. सर एक आपने पूछा था इंट्रोपी पे सवाल आपने एक पूछा था. सर प्रोटीन
एंट्रोपी पे,
की.
एंट्रोपी दैट मीन्स रैंडमनेस. ये मेजरमेंट करता है रैंडमनेस को. किसी भी सिस्टम में जितना ज़्यादा रैंडमनेस होगा उसकी एंट्रोपी क्या होगी, उतनी ज़्यादा ही होगी. तो मैंने आपसे सवाल पूछा था कि प्राइमरी, सेकेंडरी, टर्सरी, क्वार्टरनरी स्ट्रक्चर्स में ज़्यादा एंट्रोपी किसकी है. तो अगर आप देखेंगे
प्राइमरी
स्टेबिलिटी को लेकर के, तो आप देखेंगे तो क्वार्टरनरी या टर्सरी स्ट्रक्चर मोर स्टेबल होता है. दैट
उसकी
मीन्स
एंट्रोपी कम
रैंडमनेस कम है,
होती
तो
है.
वहाँ पे एंट्रोपी की वैल्यू क्या होगी, कम होगी. सबसे ज़्यादा
यस सर.
अनस्टेबल कौन है, प्राइमरी स्ट्रक्चर. दैट मीन्स देयर विल बी
सबसे ज्यादा.
हाईएस्ट एंट्रोपी. होपफुली
अब
क्या हो,
आंसर आ गया.
या.
यस. थैंक
ओके.
यू सो मच.
सो नाइस टू इंटरेक्ट यू विवेक, आपसे अच्छा लगा बात करके, एंड होपफुली कभी आप भी काफ़ी कॉन्फिडेंस फील कर रहे होंगे. ये
हाँ,
वीडियो
अच्छा लग
आपको
रहा
मिल
है.
जाएगा. जो भी सजेशंस और जो भी क्वेश्चंस मैंने आपसे पूछे हैं, उस तरीके के क्वेश्चंस को थोड़ा स्टडी कीजिए, चूंकि आप बायोकेमिस्ट्री के कैंडिडेट हैं, तो आपका बायोकेमिस्ट्री थोड़ा अच्छे से स्ट्रांग होना चाहिए. ओके.
जी सर.
कब है आपका
जी
इंटरव्यू?
सर. 13 को है सर.
चलिए,
13.
आपको आपके आपके इंटरव्यू के लिए ढेर सारी बेस्ट विशेस. अच्छे से आप
जी
इंटरव्यू
सर.
दे करके आइए. और बाकी अगर आपको कोई फर्दर कन्फ्यूजन या डाउट रहेगा तो आप मैसेज करके उसको पूछ सकते हैं.
ड्रेसिंग सर सही है सर. तो ड्रेस जो मैंने पहना
हाँ,
सही है
ड्रेस
सर.
ड्रेस आपका बेहतर है, कोई दिक्कत नहीं है. थोड़ा-सा आपके जब आप आंसर्स करते हैं, तो ऐसा लगता है कि थोड़ा आप पॉज़ लेते हैं, आपकी आवाज़ थोड़ी रुक-रुक के आती है. आई
ठीक
डोंट
है.
नो कि ये नेटवर्क का इश्यू था या फिर ऐसा आप बोलते ही हैं. अगर वो नेटवर्क का इश्यू है फिर तो ठीक है, अदरवाइज़ आप कोशिश करें कि थोड़ा अगर कंटिन्यूएशन में बोलेंगे तो अच्छा रहेगा. ठीक है?
ठीक है. ओके सर. और
ओके.
सर कोई फीडबैक सर. सर और कोई
और
फीडबैक
दूसरा
सर.
चीज़ ये है कि जब आप इंट्रोडक्शन अपना दे रहे हो, तो इंट्रोडक्शंस को 4 से 5 बार प्रैक्टिस कर लीजिए. बिकॉज़ इंट्रोडक्शन
ओके
ऐसी
सर.
चीज़ होती है जिसको बहुत फ्रीक्वेंटली बोला जाता है एकदम नॉर्मल तरीके से. उसमें ऐसा
ओके सर.
फेशियल एक्सप्रेशंस ना बने कि आप रट करके आए हुए हैं और आप ट्राई कर रहे हैं उसको बोलने का, और आप सोच
ओके
रहे
सर.
हैं. तो हो सकता है कि
सर.
आपने ऐसे कहीं इंट्रोडक्शंस अपना दिया नहीं होगा. दैट इज़ वाई आपको थोड़ी वो आ रही होगी. जो पार्ट आप
ओके
बोलना चाहते हैं इंट्रोडक्शंस में, उसको आप 4 से 6 बार ऐसे बोल दीजिए. अपने फ्रेंड को सुना
सर.
दीजिए, अपने घर में कोई छोटा भाड़ा बड़ा भाई बहन उनको सुना दीजिए, लेकिन 4-6 बार बोल के प्रैक्टिस कर लीजिए. एक बार क्या होगा कि
ओके
अगर
सर.
आपके माइंड में वो सारी चीज़ें क्या होंगी, फिट हो जाएँगी ना, तो फिर आप अंडर प्रेशर भी अच्छे तरीके से आप बोल पाएँगे. अदरवाइज़ होगा
ओके
क्या कि
सर.
जब प्रेशर आपको मिलेगा ना वहाँ पे इंटरव्यू में, तो आप उसको सही से बोल नहीं पाएँगे और आप पहले ही वहीं पे डिमोरलाइज़ हो जाएँगे, जो कि ठीक नहीं
ओके
होगा.
सर.
ठीक है?
ओके सर. ओके सर. थैंक
ये
यू सो मच सर.
ओके, इन दोनों चीज़ों को इनकॉर्पोरेट कर लीजिए, बाकी सब चीज़ें ठीक हैं, कोई दिक्कत आपको नहीं होने वाली. ठीक है?
थैंक यू सो मच. थैंक यू सर.
ओके,
थैंक यू सर.
मोस्ट वेलकम.
थैंक
गुड नाइट.
यू.